So it is my pleasure today to introduce Inigo Martínez Solano. Inigo has recently joined the Doñana Biological Station. He's been part of the phylogeographer Globe project, and he got his degree at the Universidad Autónoma de Madrid a while ago. Then he postdoc at the University of Berkeley in California, then moved on to Connecticut, changed coasts. He then took Ramón y Cajal at the IREC Institute, and then afterwards he moved to Portugal, to Seville for about a year, and he's now joined us for a little while. He's got a reputed phylogeographer for amphibians, as you will see, and has over 40 publications. He's directing to doctoral students, and he will soon start with a few more here in Doñana, right?
Hopefully.
So,
well, thank you very much for the introduction and for the opportunity to talk here today about my work. And today I wanted to talk about phylogeography, about the work that we have been doing in our group for the past 5-6 years. And it's mostly unpublished work. It's work in progress, so any comment or suggestion is more than welcome. So with this in mind, I wanted to talk about our approach to phylogeography, which involves both a comparative and an integrative approaches and a multi-scale, which I think is important to highlight because it brings additional explanatory power to phylogeography. We are all familiar with phylogeography by now. It's the study of historical processes that are responsible for the geographic distributions of individuals or historical lineages or species. And since it's about a couple decades old, it's a young discipline, but it's already old enough that there's been time for reflecting upon its contributions and avenues for further research. And I think one of the main conclusions that is that we have made much more progress in documenting patterns, and by patterns I mean identification of monophyletic groups or haplogroups, or describing the geographic distribution of these haplogroups, patterns of allopatry or sympatry, or estimating divergence times based on genetic distances. But comparatively, we have made much less progress in understanding the processes behind or generating those patterns. We know much less about the relative roles of genetic drift versus natural selection in generating those patterns. We know less about patterns of gene flow and its relationship with migration or dispersal patterns of organisms, and much less about patterns of admixture or hybridization and their role in the speciation process. So this is a problem because if all we have to infer evolutionary processes are patterns, there are a variety of factors that can mislead us, and these include biases in sampling design or in the choice of molecular markers, using fast versus slowly evolving markers, for instance, or with the assumptions you make, like equaling gene trees to species trees is a hot topic these days. So, but basically the idea is that similar patterns can be generated by very different processes. So we need to gain additional insights about these processes. And some of the proposals that have emerged relatively recently involved or proposed that phylogeography should be more statistical, or that there should be a better integration of geographic information systems in combination with molecular data to improve the explanatory power of phylogeographic inference. and this has been achieved, and these are very active lines of research. But I argue that there is still a pending connection, which is the demographic connection. Some years ago, David Buckley published this essay reflecting on the past and the future of phylogeography, and he argued for an organismal, integrative, and iterative phylogeography. And we can probably agree that phylogeography is integrative, is using more and more different sets of data, and it's also, thanks to these more rigorous statistical approaches, it's become an iterative discipline that progressively refines new hypotheses based on previous results. But I don't think it's the organismal component is so well integrated into the inference process in phylogeography. And in this paper, what David was arguing for was that we need to integrate more of the life history of the biology of the organisms, that they should be central to phylogeographic inference, and usually that's not what we see. So how do we plan to fill this gap or to contribute to this? Well, we have been working at 3 different scales, and we hope to obtain new knowledge at each of these stages that will inform the other different scales, and this will not only refine our hypothesis and produce more robust hypotheses about the evolutionary history of the organisms, but we will also be able to hopefully integrate this information from the organisms themselves. So the 3 scales at which we work, starting from the larger to the smaller, the first would be the classical, the range-wide, the phylogeographic inference approach, including use of or the estimation of time-calibrated gene genealogies, applying phylogeographic diffusion analysis to understand patterns of expansion, use of Bayesian clustering algorithms to understand range-wide patterns of genetic structure, and use of species distribution models. And I will put some examples later. At the next level would be a more regional level, roughly what's called landscape genetics, including estimates of fine-scale patterns of gene flow, analysis of isolation by resistance, and estimation, integration of these estimates with estimation of effective population size. In amphibians, this is very useful also in conservation, and I will put some examples also. And finally, the local scale would be focused, or would focus on life history traits and the demography, and with things like the estimation of the ratio between effective and census population size, or estimation of actual dispersal distances of marked individuals. I'm going to illustrate some of these stages using 2 study species, which are a salamander, the ribbed newt, Pleurodeles waltul, and the Iberian spadefoot toad, Pelobates cultripes. And these 2 species are not strictly Iberian endemics, because in the case of, they have very similar distributions. They overlap in extensive areas in their ranges, and they are often syntopic, which means you can sample both species in the same spot. There are some differences, like you can see Pleurodeles has some populations in North Africa, which origin is uncertain and is debated. And Pelobates, there are some populations along the Mediterranean and Atlantic coasts of France that have also been the subject of particular studies. These two species have an old presence, they share an old presence in the Iberian Peninsula. There are Pleistocene fossils of both species, and actually some of the oldest dates for the species in the Iberian Peninsula come from the same fossil site, so you could argue that they are syntopic, they have been syntopic for a long time. We're talking about 2.6 million years, and there are records of the species in Atapuerca, for instance. And both species have sister species in North Africa, so they share some things, including part of their evolutionary history, but they differ in some other respects. And by studying both of them, we expect to learn some things about both about generalities and about factors that specifically affect one or the other. This work I am going to talk about is a long collaboration involving and is part of the PhD projects of two students, Jorge Gutiérrez, who is in Madrid at the Museo Nacional de Ciencias Naturales, Goyo Sánchez at the Universidad de Navarra, and our collaborator, the postdoctoral researcher Marcia Barbosa, who is in Universidad de Évora, and she's the expert in modeling. When we work at this range-wide scale, our major goals are to well, the typical estimate the timing and the geography of diversification, and to also identify climatically favorable areas through time for both species. Because we want to infer whether these areas really function as refugia, and that means that they were reservoirs for genetic diversity in these species. We emphasize sampling in our research because we think it's very important, especially when you want to use some analytical tools. It's important that your sampling is comprehensive and it's balanced. And so we sampled Pleurodeles, for instance, in over 50 localities throughout the Iberian Peninsula and in North Africa. And we used both mitochondrial DNA and polymorphic microsatellites that we have specifically developed for this project. When we look at mitochondrial DNA, we find a lot of structure in Pleurodeles. We have an old split about 3 million years ago between eastern and western clades. The western clade is very widespread throughout the western half of the Iberian Peninsula, but there's also some divergent haplotypes in the southwestern region of Portugal, which is in accordance with results in several other amphibian species and in reptiles. And in the eastern clade, we don't have good resolution for some of these clusters, but I wanted to highlight in particular this cluster of haplotypes that is exclusively found in Morocco, and that in principle would argue against a very recent colonization of the species in North Africa, as some authors have proposed. The genetic distance is quite important, although in amphibians it's not big, but it's over 5%. And with microsats we have a similar picture, but there are some contrasting results, and in particular I wanted to highlight 2 or 3. The first is that this divergent group of haplotypes from Algarve, this group is not recovered with nuclear DNA, and we also don't recover any distinctiveness of Moroccan populations. And instead we find this group of populations north of the Sistema Central Mountains that is recovered as a genetically differentiated group at this scale. These discordances are interesting actually, and give us some clues about the evolutionary history of these populations. In the case of North African populations, we have exclusive mitochondrial DNA haplotypes, but no detectable differentiation at microsat loci, and we are running analysis with the isolation with migration model, which suggests that rather than a single colonization event, there was a period of time in the late Pleistocene where there was bidirectional migration between southern Iberia and North Africa, which is interesting because it's not often the case that we see this pattern in amphibians, for which the Strait of Gibraltar tended to act as a very strong isolating barrier. On the other hand, this group of populations from the northern end of the Sistema Central Mountains, we think they are recovered as distinct because of a demographic effect, of a founder effect, and in fact, structure. This software is known for being sensitive to this kind of founder effects when recovering population clusters. Additional evidence comes from the phylogeographic diffusion analysis with software BEAST. You can see here, well, it's not a great figure, I agree, but what you have in different colors would be inferred ancestral areas for the western and eastern clades, which, by the way, the colors are reversed, I apologize. But in lighter colors are populations that are inferred to have expanded or to have colonized more recently. So northern populations within both clades are inferred to have colonized these areas more recently, and according to the reconstruction of effective population sizes in the vertical axis through time, estimate these demographic expansions in the last interglacial period, around 100 or 120,000 years ago. When we look at the models of favorability for the species in the present and in the past, we see that there are big fluctuations in favorability for this species, with a strong reduction of favorable habitats in the last glacial maximum. And we can use this information to look at what areas were climatically stable through time, at least in this period of time. And when we map that, this area in the southwest is identified as the most favorable area through time in the species, and this correlates very well with measures of genetic diversity. So this is good evidence that this area was a historical refugium for the species and a reservoir for genetic diversity. In this figure, we have the overlap between the climatically favorable areas through time, and here what we see is the first principal component of 4 genetic diversity indexes, including mitochondrial DNA and microsats. And you see there's visually you see there's good correlation. You can trust me that there's also statistical correlation between the two maps. What happens in the other species we are studying in Pelobates? Well, as is typical in amphibians, and in particular in the Iberian Peninsula, there is also a good degree of population structure at the mitochondrial DNA level. But as you can see, this structure is much more shallow. This would be a network of the haplotypes we find. There's more admixture, and genetic distances are much lower. This gives us much more recent estimates for time for coalescence of all these haplotypes, and coalescence would be within the Pleistocene, within the last million years. In this case, we also sequenced the same mitochondrial DNA marker, and we also used a set of polymorphic microsatellite loci. And again, with microsats, we see a structure. We see much more widespread patterns of admixture across groups, and we see that northern populations tend to be recovered as differentiated, but this is also probably a result of demographic effects, as I will show. In this species, we don't have big fluctuations in favorability through time. You can see that they are not, or changes are not as marked as in Pleurodeles. And you can also see that this would be, again, the map of genetic diversity for the species. And apart from more northern populations, which always are recovered as genetically impoverished, you can see that the genetic reservoirs were larger than in the other species. So to conclude with this part, we have seen, or we have evidence for differences in the timing of diversification between the two species and about in the geographic extent of the inferred refugia. We also have inferred slower diffusion rates for Pleurodeles. This is one of the parameters you can estimate in the continuous diffusion models in bees. It's called the, here we go. It's called the diffusion rate, and it's on a scale of kilometers per million years. And of course, it's conflating things like the evolution of climatic favorability through time with the actual dispersal ability of the species. But so this difference could result from both factors, and not necessarily one or the other, but probably in combination. But it's an important thing to keep in mind when for instance, we look at or we build models to predict where the species will be in 2050 or 2100. These values are really low, very slow. We have documented important demographic founder effects in northern populations, which are genetically depopulated, and this is important also from a conservation perspective. We can see that populations in France probably result from a rapid colonization event, after which there was a fragmentation, but they are genetically almost indistinguishable. So this goes a bit against my previous slide, but there's certainly differences in the expansion or in the diffusion rate through time. So in this species, in Pelobates, at least, this seems to provide good evidence that given the right climatic conditions, they can indeed disperse relatively rapidly. But that's probably not the case in Pleurodeles. This is just a comparison of the patterns of genetic diversity in the two species, just to highlight once more this north to south trend in genetic diversity. And we have seen that populations north of the Sistema Central Mountains tend to be genetically differentiated in both species, and that this probably results from a founder effect. So these mountains seem to have acted as a major barrier to dispersal. We can further test this at the next scale of analysis, which would be the what we call the landscape scale. It's not really a landscape, but here we want to, for instance, to evaluate the role of these mountains in shaping patterns of gene flow through time. This is a comparative approach involving more species than the two we are talking about today. This is work in progress, but I present some preliminary results here. Apart from the role of the mountains on themselves, we would like to identify other landscape features that may promote or restrict gene flow in the two species, and also characterize regional population networks that would be characterized by effective population sizes at the breeding site level and the patterns of gene flow with neighboring breeding sites at this scale. This would be a map of the eastern part of the Sistema Central, the Guadarrama Mountains in Madrid. This is a typical sampling for some of the species we are studying, and the idea or the null hypothesis here is that patterns of gene flow will be higher when you are comparing populations on the same slope than when you compare populations from different slopes. We have mostly data only from the southern slopes, though, but I will still show some results. This would be this southern part, and this is I don't know if you can see very well, sorry about that. The sampling at this scale is also, we try to sample, when possible, the two species at the same spot. If not, trying to find the most or the closest population in order to have a balanced sampling in terms of number of locations, individuals per location, average distance, geographical distance between locations, and also the markers we used. Of course, they need to have comparable levels of polymorphism in order for comparisons to be meaningful, actually. So we do find some differences in the regional genetic structure of the two species. And in Pleurodeles, typically you find good support for higher clustering levels. So, for instance, between 8 and 12 clusters out of a sample of 20 populations. Only populations that are really close together in the order of under 10 kilometers will be recovered as genetically similar, but farther from those distances, they show detectable differences. In Pelobates, it's not the case. You tend to recover values, very low values of number of clusters, and probably 1 or 2 clusters, even at this scale of analysis, is the scheme that best explains the. Here is a comparison of the average distances between populations and FST and DST values, which are relatively big in Pleurodeles, but much lower in Pelobates, especially when we look at DST values. This would be a plot of our estimates of effective population sizes in the breeding sites we sample, and they tend to be higher there is a lot of variation across sites, but values tend to be higher in Pleurodeles than in Pelobates. So these differences we find at this level, preliminary as they may be, may help explain at least part of the difference we find at the phylogeographic level. Why do we find much lower, much shallower population structure in Pelobates than in Pleurodeles, which, by the way, is also a typical pattern when in amphibians you compare salamanders with frogs. Salamander populations also always tend to have more structured populations. This is a reminder of this. But this is a genetic estimate, so if we go to the next level, we can ask what is the relationship of these estimates with the census population sizes, and whether our genetic estimates are accurate or can be calibrated, for instance, in the case of Pelobates with the counting of egg masses or other approaches. So at the final scale of analysis, our goals are to estimate census sizes and effective population sizes, and as well as the ratio, which is the relevant parameter on an evolutionary timescale, and also to quantify typical dispersal distances in the two species. So this is our study area in the mountains of Guadarrama in Madrid, and we tried to choose an area with no internal barriers to dispersal. So this is not divided by roads or anything. It has two major breeding sites for the two species that are about 700-800 meters distant, and a network of trails. So during their breeding season, we do intensive mark recapture sampling sessions in both the breeding sites, and out of the breeding season, we monitor these trails in order to try to document possible dispersal events. This gives us certainly an underestimate of dispersal distances in the species, but logistically, this is the best we can do. I'll show you some of the results. This is just for you to get an idea. These two sites are very different in hydro period and about their origin. One is a natural pond. It's an ephemeral pond that dries up. It fills during the winter, and usually by May or early June is dry. And this is an artificial pond that is much deeper and keeps water throughout most of the summer. What we usually do is just during these sampling sessions, we just, every individual that's collected, we take biometric measures, we sex them, we implant pit tags to identify the animals, and we take tissue samples for skeletal-chronological and genetic analysis. We record the location of the animals. The idea is to contrast or to compare field estimates for census size or for dispersal distances or for the sex ratio with genetic-based estimates like the effective size or gene flow, or we are interested in inferring patterns of sex bias dispersal, since dispersal is hard to record directly in the field. Maybe we can infer through genetic methods and see if there are patterns of sex bias dispersal as has been proposed for different species of amphibians. These are some results based on 5 years of marking. We have captured over 1,000 individuals of each species, corresponding to almost 700 unique individuals in Pelobates and almost 900 Pleurodeles. Recapture rates overall are close to, well, are higher in Pelobates, almost 46%, 36% in Pleurodeles. It's not that great, but this is because we are including juveniles in the count, which are much harder to recapture. And when we look at the distribution of dispersal distances, we see that almost all of the events correspond to small scale. This would be within the buffers around each of the breeding sites. So most of the times we find every animal in the same breeding spot. We do find some dispersal events that seem to be a bit more frequent in Pelobates than in Pleurodeles, as was our working hypothesis. But unfortunately, the number of dispersal events that we have recorded in this period of time, despite considerable effort, is not good enough to make any robust conclusion. But this will be our census estimates. Some years we've had trouble because of climatic conditions. 2012 was a very dry year. There was no breeding in Pelobates at all. We did get some we could catch some pleurodeles, but these are partial data. But based especially on data from 2011, 2013, we can estimate adult populations of 430, 75 pleurodeles, 300, 360 pelobates in both cases with male biased sex ratio. We have tried to infer effective population sizes with different programs. I'll report the results with Colony that we think is providing the most accurate estimates, and for these estimates we assumed monogamy in both sexes, and we used 21 microsats in pleurodeles, 12 polymorphic in pelobates after exclusion of markers with disequilibria or potentially problematic. And we, in both species, we genotyped a larger sample of larvae than to make sure that all animals were from the same cohort. But we also included a sample of males and females from that population that you can specify as potential parents, and that is supposed to, and in pelobates it does provide more accurate estimates of effective population size. The ratios are relatively high for amphibians, which typically or frequently have ratios between 10%. But in these two species, they seem to have similar or comparable ratios. So what we found, inconclusive as it may be, the low number of dispersal events and the low distances that they covered certainly explain at least part of what we see in the genetic structure, both at the regional and at the phylogeographic scale. These values, well, they need further analysis to test additional software, and we are worried. For instance, we have been using software OneSump to estimate effective population sizes, and we have found differences across replicates, differences depending on the set of priors you use in the analysis. So this needs further testing. But with pelobates, we have found good agreement with NB estimates produced by Colony and our counts of egg masses. Unfortunately, with pleurodeles, you cannot calibrate your genetic estimates because they lay eggs individually, and you simply cannot follow a female newt and count how many eggs she lays, apart from other logistic problems. But with pleurodeles, with pelobates, you can count the eggs, and the agreement is not bad. So maybe we can generalize our protocol to study of or to the monitoring of pelobates populations in different systems. To finish with the presentation, what have we learned? Is it worth to make this multi-scale approach, and well, or the comparative part, is it valuable to compare different species? Well, we have found similarities that could be related or that in principle are easier to assimilate to historical factors, but we have found differences that are, or at least for us, were unexpected when we started the study, related to timing of diversification, to the diffusion rates inferred for the species, about climate favorability. And I think the benefit of our approach is that you can, at least to some extent, separate the relative roles of historical events, like vicariant events, or landscape and life history related factors in explaining the patterns of population persistence and divergence, which eventually is telling you about the evolutionary history of populations or the species you are interested in. As a side result, I think these estimates of effective population sizes and the regional patterns of gene flow, they set the basis for the long-term genetic monitoring of amphibian populations. Once you have characterized network of breeding sites through their effective population size and connectedness with other breeding sites, you can identify which populations have more leverage on population persistence at this scale. So it could be useful to work this out and design programs that with a sampling period every 5 or 10 years would give you a good estimate of both abundance and connectivity of amphibian populations at a relatively large scale. And if this is generalizable, one great system, and I'm very interested in testing whether we can use all this information in Doñana. Doñana has a rich amphibian community, is well known for decades, and there are gradients of temporality and connectivity between the breeding sites that make it an ideal system to test some of these results we have obtained in different systems. And we are now we have focused on 2 species with very divergent life history traits. In the case of Pleurodeles, we have seen that it's a very philopatric species. They don't move a lot. They have very marked structuring. In the same area in Guadarrama, we were also studying green frogs, Pelophylax, Pereci, and they are the other extreme. They move a lot. Even within the single breeding season, you will recapture animals in different breeding sites. They move much more, and they have no genetic structure detectable at that scale. So we are interested in comparing the 2 species, integrating well, the molecular tools and life history information in this context of the gradient of temporality of the ponds, how the 2 species adapt to breeding in ponds that unpredictably vary in their hydroperiod, in the time available for development of the larvae. This is work that will be part of the PhD thesis of Mark Comas, who recently got a predoctoral fellowship from the Severo Ochoa program and will be co-directed by Polly, who's around, I hope. And the idea is to use the molecular markers we have optimized in our study populations and combine our genetic estimates with demographic estimates in the field, use of skeletochronology to characterize the age structure of populations breeding in different conditions, and combine this with experimental approaches to try to understand the basis of adaptation of the 2 species in this gradient of temporality. And hopefully that would be the subject for another talk in the future. If you are still interested, I hope you will come. Thank you for your attention, and if you have questions.